Thank you. Thank you for joining us today. I hope everyone is logging in effortlessly. My name is Dr. Neda Gioia. I'm the president of the Ocular Wellness and Nutrition Society. And our goal is to bring worldwide evidence-based nutrition and wellness strategies to healthcare providers, scientists, public, to help change the course of vision loss and poor health. We're passionate about saving sight and providing an unbiased representation of well-designed studies. OWNS is a 501c3 charitable nonprofit organization, and we're privileged today to welcome a distinguished speaker, Dr. Amine Zorgani, to get into a topic of the microbiome, which is a complex network of microorganisms that play a crucial role in our overall health and well-being. Understanding the microbiome is more than a scientific curiosity. It's a necessity for anyone interested in maintaining and enhancing their health. Whether you're a healthcare professional, wellness enthusiast, or simply looking to improve your own health, this webinar will equip you with some advanced knowledge about the topic. Please use the Q&A box for the last few minutes of the session, as we will pluck some of your questions for Dr. Zorgani. And we're thrilled to welcome LKC as our newest gold sponsor. Your support is invaluable to us, and we're excited about the opportunities this partnership brings. Thank you for believing in our mission and contributing to OWNS. And as I said before, we're a 501c3 nonprofit organization. Our growth is sustained through contributions of our members, donors, industry supporters, sponsors, and volunteers. By joining as a member, you contribute to the momentum that drives our educational initiatives within the dynamic and evolving realm of nutrition and lifestyle medicine and eye care. Some recent membership benefits include our doctor lookup feature, where you can connect to patients and colleagues who are passionate about lifestyle medicine, our ongoing collaboration with the American Nutrition Association. Many of our graduates are already applying their knowledge in this whole body nutrition education, thanks to this new partnership. Please be advised that OWNS members receive the highest tier discounts in this program. Make sure to sign up before this offer expires at the end of the year. And we're always enhancing our lectures with COPE approvals. And I'd like to save the date for you to Monday, May 13th with the wonderful Dr. Caleb Abbott, who will explore the relationship between sleep and eye health in our next exclusive COPE lecture. For non-members, select COPE lectures will be available for a fee, including Dr. Abbott's, which has recently been fully COPE approved, so sign up soon to gain that access. Okay. Now I can start my formal introduction to Dr. Amine Zorgani. He is a scientist and microbiome expert with over a decade of experience working with biotechnology startups on both gut and skin microbiome. He dedicates his life's mission in saving the human microbiome from extinction using his Microbiome Mavericks Communication brand. Dr. Zorgani works with scientists across the globe to bring microbiome science to the wider audience and bring innovations from the bench to the bedside. I am deeply honored to present Dr. Zorgani today and his expertise in this subject matter that I not only professionally believe in, but I also have personally improved my own health with looking at my own gut health. I thank you, Dr. Zorgani, for accepting this invitation for our audience, and I expect much enlightenment and interest going forward. So without further ado, I'm going to give Dr. Zorgani a few moments to screen share. And as I said before, our Q&A will be at the end of the session. So thank you.
Hey, Neda, thank you so much. And I'm very pleased as well to be here. So just do you hear me properly? Because I just put my earphones on.
Great.
Okay, cool. And the slides are also shared. No, thank you so much. And really pleased to be here with you. And thanks for who joined us to this lunch or probably evening if you are in Paris or around the European zone as well. So what I try to do as well today is really give you more of a microbiome 101 in the beginning and really try to, how to say, excite you as much as I'm excited. And if I get you hooked on the microbiome, that's all what I want. Because I always tell people the minute you get into the microbiome field, you will never be able to get out of it for the simple reason microbes are everywhere on you, around you, in you, and you see them or you don't see them, but they see you. So just be careful of that. This is a real email that I'm gonna read for you here, and there are some, how to say, some good things and some bad things about it, right? So, dear doctor, I have discovered when what none of my previous doctors could tell me about my body. I paid for an expensive microbiome test at a private laboratory and found that my microbiome is abnormal and damaging my health. I am convinced this is causing of all the medical problems I've suffered from since childhood, which the doctors could not diagnose. I urgently need a fecal microbiota transplantation, FMT, to get well. My friend will be the donor. Please arrange for me to have the FMT. So the good things about this, that this lady had mentioned the microbiome test, which is a new innovation that today we can literally, using the technology of microbiome sequencing, look at the microbiome of people. What is it inside? What's composed? What are the bacteria that are in there, or what are the bacteria that are missing? And what so? And abnormality of the microbiome is what we call also dysbiosis. So when the microbiome is imbalanced or dysbiotic, that's the abnormality that this lady is mentioned here. She's also saying that she's convinced that it's causing all of her medical problems that doctor did not see. Although being convinced of something needs to have proof. Today, there are more than 360,000 papers published in the microbiome, and they touch multiple diseases, either they're communicable diseases or non-communicable diseases. Probably the convinced part of this email is because she found a lot of paper basically saying that her condition is related to a microbiome dysbiosis. However, the how to say, the bad thing, or probably the presumptuous thing about her email, is that she thinks immediately that because the microbiome is dysbiotic, that she immediately urgently needs a fecal microbiota transplantation from another person, which is her friend. This is not how we do fecal microbiota transplantation, and it's not just because someone did a microbiome test. It needs a lot of more investigation for someone to actually get a fecal microbiota transplantation, and you don't get it from a friend like a DIY. So this is how to say, how nascent is the field of microbiome. So there is so much research and there is so much excitement to the point that people are ready to really risk it all and get a fecal transplant from someone, friend or a husband or what so, because they've tried everything and it didn't work. And the microbiome seemed to have that solution. The microbiome seemed to be that immune medicine. And this is not like just random cases. This lady, she had a car accident, Caitlin, and she was suffering from C. diff. And this is published like in 2012 in the CNN. This is just one case. And she did everything and she couldn't get rid of her Clostridium difficile infection, which is a recurrent infection. And she got a fecal microbiota transplantation from her mother, and she was recovered very quickly right after that. This is, again, not an isolated case, that this fecal microbiota transplantation does work. However, it is very important to keep in mind that information without a perspective is a higher form of ignorance, because you need to always keep a perspective on what is a good science, what is the science in the microbiome field that we can actually today use, because there are two groups. There are the group who tells you the microbiome science is really good and there is a lot of application. There are also the other side who tells you, that's not a science. I mean, probably they've discovered something, but we cannot use it yet because these microbiome tests aren't accurate anyway, and people that are selling them for marketing. But there is the middle one. There is the middle one of the science. They really use the science that is existing today and try to leverage it for applications. And this is really what I try to show you today, step by step. What is the microbiome and how is leveraged and how you can potentially use it in your medical practice or use it in your own health as well. But to start speaking about that, we need to get a perspective. okay, who's actually known about the microbiome? This is a very interesting, how to say, survey that the Biocodex Microbiota Institute did on 6,500 people across 6 countries, including the United States, France, China, and many others. And they found really very good, interesting results. And I really advise you to read this report. It's called the International Microbiota Observatory from the Biocodex Microbiota Institute. One of the first things they found is that 1 in 5 people, 1 in 5 on these 6,500 people knew what a microbiota meant anyway. Only 24 people said they knew exactly what a gut microbiota is. And when it comes to the other microbiotas, looking at the vaginal microbiota, only 18% knew, knowing that we have half-half, right? So if you consider that there is half women, half men, only 18% knew what a vaginal microbiota is. Same for the oral microbiota, very little, and pulmonary microbiota. Now, these are not the only microbiotas we have. There are many other microbiotas. We're just speaking about before this, and this is the Ocular and Wellness Nutrition Society, the eye microbiota. It does exist, but we don't speak about it, and probably people don't even know it does exist. And then there is only 1 in 4 people have heard of the term dysbiosis. So if you don't actually know what a dysbiosis means, can you even know it exists, and how can you link it to your condition or your patient's condition? And the other hand, which is very interesting, 2 in 5 people have been informed by their healthcare professionals of the appropriate behavior to adopt to preserve their microbiota. This is the only small proportion of people. What is very interesting, and this is the trust relationship that the patients take with their doctors, right? So patients trust their doctors. So when a doctor informs their patient about the importance of their gut microbiota or their microbiota in general, in 95% of cases, these people took some action to actually do something about it, to keep their microbiota healthy. So because of the trust they have with their doctors, they listen to them. What does this mean? Meaning that we need to increase the education towards healthcare professionals about the microbiota for them to communicate appropriate information to their patients to allow them to actually take action about it. And this is really my mission. This is really why I accepted to be here today, why I was honored to be here today, is really to inform, educate about the microbiota. And this is what this microbiota is. This is the collection of these microorganisms. Is not just bacteria, although we tend to speak a lot about bacteria, but microbiota is a community of microorganisms that include bacteria, includes archaea, which is a higher form of bacteria. It includes viruses. It includes yeasts and fungi in the environment. And the microbiome, which is the collection of these microorganisms and what they produce, what they do in their environment. And this is my mission. This is my life as well. I've been working with microbes for more than 19 years now, to the point becoming an influencer. This is the word that I didn't want to use, but people actually put it on me. They said, listen, you are an influencer. You should say that and should show it, because we actually need influencer in this field. I'm showing it here because I really want to inspire others to speak up about the microbiome and just at least reduce some complexities that people might have in there, and being an ambassador as well. I'm also trying my best to kind of, how to say, be also a spokesperson at the highest institutions. Here was an European Commission, where we're really trying to actually define what a microbiome means in a food environment. So because microbiome is not just for humans, it's also for food, plants, and micro animals, and the environment itself. And part-time inventor, that's a different thing. But I'm a scientist before everything. I've been working with startups mainly, both either using AI in the microbiome, gut microbiome, or being hosted in the Johnson & Johnson Innovation Labs for 3 years as a director of R&D for another scale microbiome company. So this is what I do for life. It really breathe in microbes. And from my communication, I'm also an author of the first Microbiome Mavericks audiobook, and I've been honored to interview many of the smart people, some you probably know, on this field, and continuing this as a podcast that I highly invite you to listen to, the Microbiome Maverick podcast as well. So now going deeper into the discussion and trying to really get you into what is the microbiome and get you excited as much as I am excited about the microbiome. Obviously, when you speak about microbiome, you speak more about bacteria as well, right? If you look into this, this is 4,000 million years ago. The first thing that happened on Earth, probably beyond Earth, is bacteria. So bacteria is the most ancient form of life existing on this planet, and probably beyond. Who knows? Maybe tomorrow we'll discover that bacteria actually existed on other exoplanets that we don't know of. These bacteria actually, on the other side, they are the second biggest biomass. If you look into the plants, they represent 450 gigatons, where bacteria represent 70 gigatons. Animals include human in this case, which only represent 0.01% of the all Earth biomass. So bacteria as a biomass is one of the second biggest biomass, which gives you must think about it. If there is so much bacteria out there, there is a reason why they're there, because the role in what they do, it's very crucial and it's very, very important. It's also that in human body, they outnumber us with 360 times. So us is literally 360 times genes less than what the bacteria would have on us. When you look into more of the bigger picture, 95% of our microbiota is located in our intestine, right? It also has this equilibrium between 1.3 times. You might find in some reports that bacteria actually is 10 times bacteria than we have human cells, but there are some other really strong calculations that showcase 1.3 times bacteria when the compared to the eukaryotic cells, which is our cells. It's in your intestine today, you have 2 kilos of microbes. These microbes are actually also your fingerprint. To some point, we might be able in the future identify people based on their microbiota, literally tell you who you are and what you probably ate yesterday, because the advance of the technology can really allow us to do that. When you look into from a data perspective, and we are living in the data world, we are living in social media where the data flows in and flows out. The human stool actually contains a vast amount of information. 1 gram contains 1 billion of microbes, each within 1 million DNA base. If you just take a spoon of stool, which has information that contains 1 ton of DVDs, imagine that how much of information you would have there into and on what you actually need to analyze that information. And for us to actually study these microbes, it didn't happen just like that. This is started in the 16th century, where the first invention of the microscope and Antoine Leeuwenhoek, who basically started looking into these microbes and just grow gradually. And then we started culturing these microbes. We actually wanted to understand, okay, are they different groups or are they just one group of bacteria? We started staining them, coloring them with the invention of the gram staining. You probably heard of it. Gram-negative bacteria and gram-positive bacteria, which they do have a different cell wall. And then with the advancement of the technologies of the sequencing, we started sequencing these microbes to actually understand exactly who they are and using this 16S sequencing. So this is which brought us to the new era of technology, using the DNA, this information that actually holds whatever the bacteria, who is there, using the 16S ribosomal RNA, which is this small, the 16S is like, how do you say, you see your ID card and they have this chip on it, that's exactly the 16S. So each bacteria has its own 16S. Then we try to sequence that 16S using a PCR. You're very familiar with PCR now with the COVID. So we do this PCR to amplify the 16S gene and then sequence it, which will give us who is actually there. We do the 18S for fungi and yeast as well. And if we actually look into bacteria in isolated samples, we do the 16S and it's easy. And if we are looking into an environmental microbiota samples, like the stool samples, which contain not just one bacteria, but contains a lot of different strain and species of bacteria, we do what we call a metagenomics, literally sequencing every piece of DNA in the sample. The second level of understanding we have, what we call it RNA-based approach. This time, the first one was in DNA was who is there. With the RNA is how do they respond? What are the responses they have, would we call them the meta-transcriptomics, because the RNA is which carries the information from the DNA, and then it translates it to RNA. And then the third level, which is the protein-based approach, we call it the metaproteomics. This is more how are they interacting with the host, which is us or the environment, and what our proteins are being produced. So the DNA give you the RNA give you the protein, and then there is the third partner coming from the DNA, which is the metabolites. And this is looking into metabolomics. This is more of the chemical interactions and how all these are interacting. The first two technologies that we're actually using to understand the variety of these communities, we call them the next generation sequencing. And stunning enough, I mean, honestly, when I started working on this probably in 2005, I thought we've actually discovered all microbes existing in the planet. I mean, we know bacteria, we know archaea, we know viruses, we know mostly all of them, until just the beginning of this year. We've actually discovered a completely new thing that we didn't even existed, and we call them obliques, right? This is what we call them, like, they look like viruses, and they were found inside a microbial cell. In this case was streptococcus. It's like an oral bacterium, which is a commensal one. But in total honesty, we don't actually know what they are doing. It's just a so fascinating field. We might be able to use them, I don't know, like drug design in the future, because they're so tiny, and they seem to be coding some smaller proteins, but we don't even know what those proteins are. This is to bring you to the reality that even though we've been working in this field for a long time, there are probably things that we haven't discovered, and there are probably things that we don't even know what they do. However, there are a lot of things that we actually discovered, and there are a lot of things that we actually know what to do. So when we look into microbiome and analysis, what we actually do with those gigatons of data that I told you about it. So first of all, we need to define what is the community we're studying. So maybe we're studying people suffering from chronic illnesses like IBD, for instance. So we take those people, and then we take another cohort of healthy volunteers. We get their stool samples, for instance, and then we sequence those ones. So once we have that information of the sequencing of their microbiota, and ideally their metadata, like their health status, their BMI, their age, their race, and their sex, and all those informations, we do a disease correlation between the microbiome that we sequenced and the host that we're studying. And this is disease correlation. And this is what most of this, microbiome non-believers do points. They say, all what you guys are showing, they're showing disease correlations. You're telling me, okay, IBD, people suffering from IBD, they have a reduced bifidobacterium population. So, but that doesn't mean that this bifidobacterium population caused the IBD. Maybe it's because of the result of the IBD itself, right? This is why it's very, very important to do causality studies. What we do, we use scientific communities. For instance, we get stool samples from healthy volunteers and from people suffering from IBD, and then we are to produce those culture conditions to some extent in a synthetic condition in the lab and try to analyze that. One of the most successful experiments, obviously, is using germ-free and wild mouse models, where we basically, this is like sterile mouse, they've never seen microbes in their lives. And then we take the first actually experiment was done in the lab of Jeffrey Garden, we call him the father of the microbiota, or the father of the microbiome, where the first thing they've done, they took germ-free mice, and then they transplanted a microbiota from healthy volunteers and a microbiota from people suffering from obesity. What happened is that these mouse actually that had received a microbiota from obese people, the mouse itself became obese, which is insane. It tells you there is a causality between these microbes from an obese person that causes the obesity itself. So once we understand this relationship, and that there is some microbes in the sample of the stool, some could be 1, could be 2, could be 100, could be a full community that causes the condition, this is where we can go and start making the intervention itself. Oh, I think I went back, sorry. So some sort of the interventions could be a dietary intervention, because obviously the microbiota, speaking about the gut here, is one of the engines that ferment these dietary interventions. We could do a pathogen or pathobiome depletion. The worst case scenario is using antibiotics, as you know, but today we are advancing into new technologies like bacteriophages, these viruses that actually stick to the bacteria, kill them, and in a targeted way. There are some companies who actually can now use the CRISPR-Cas technology, this molecular scissors, to actually modify the bacteriophage and make it very specific to target small set of bacteria in the gut, leaving all the other microbiota healthy. So you don't have to use antibiotics to actually destroy all the microbiota to fix it. So destroying microbiota to fix it is really counterintuitive. Using precision probiotics that actually can increase the diversity of the microbiota. Postbiotics, which is the postbiotic is the product of these bacteria and the probiotics. And the whole community transfer, which is spoken about very briefly, this fecal microbiota transplantation. And I will detail it a little bit later on as well. So these are the level of intervention. But to be able to do those interventions, you have to understand how this microbiota is actually shifting. The microbiota is not stagnant, as it just evolves with you and it reacts with you to some extent, that if you eat a burger now and a Coke, expect your microbiota to shift slightly after 24 hours, right? The first microbiota you have is called the first microbial flux that you get. This is when you get born. If you are born with natural birth, the first microbes you see are the microbes of the vaginal microbiota of your mother, where you get this microbial colonization that allows you to go those microbes that first contribute to your first birth of microbes. And then these microbes start actually, this is what we call the alpha diversity. This is the variability of the microbes throughout your age in the same person. And you can see it start increasing to stabilize on the adulthood. So your microbiota is not that stable in the beginning. Like it's very, how to say, fragile. So you really don't want to harm it with antibiotics. You don't really stop breast milk. If you can give a breast milk, give it, because there are many studies showing that breast milk is better than infant formula in terms of keeping that diversity and increasing what we call them the bifidobacterium, is the first microbes that you get with E. coli that disappears afterwards. Your microbiota is more stable, is more robust, how to say, even resilient when you are adult, but it starts getting a little bit weaker with the age, because these are age-related illnesses that comes with the wave, which also correlates with the immune senescence, because the immunity also gets a little bit weaker at the age. The microbial diversity, as you can see, this is the beta diversity. Is it between different people, right? It's very variable in the beginning, because people, they're born in different ethnicities. Some of them, they're born C-section, some of them not, some breastfed, some of them not, and so on. And then they get closer to some, how to say, a common microbiome. Although I have to mention that you are somewhere in the world now. I am in France. My microbiota is very different than yours. Although we do share some keystone species, I'm probably, eating more, how to say, a Mediterranean diet when I was living in the north of Africa. While if I get somebody living in Japan, eating sushi and rice and algae, they must have those microbes that enables them to degrade what they're eating. So there are some keystone species, they're not variable, they stay with us, but the other ones, they adapted. This is what we call the environmental microbiota, which shifts with your diet, with your location, with your age, with your wherever you are. And there is the engrafted microbiota. This is the first thing that you get when you're born that stays stable with you. We will call them also the keystone species. They stay there and they kind of help us to get through our life as well. So you really don't want to harm them. This is a very small example and how this bacteria actually changes. As you can see here, every date here represent a microbial gut testing. So this person, this is what's from the Rob Knight lab, basically took a microbiota test, and this is what you can see here, like the diversity. How diverse is the microbiota, the gut microbiota of this person? And you can see clearly that when the person actually did, when this person actually did colon cleansing, like his colon was clean, you can see that the microbial diversity drops immediately. But what's important that this microbial diversity goes back to normality after few days, which still tells us this microbial diversity is a resilience of the microbiota. Meaning, if you actually, by some reason, have to take antibiotics because you don't have a choice, sometimes you actually have to take antibiotics. And so if the doctor recommends to you, do take them. But you need to keep in mind, if you are healthy, your microbiota will probably get back to the normality. And that's most studies actually, there was a meta-analysis that showed that the microbiota goes back to normality after some time. However, if you have suffered from a microbiota dysbiosis in your earliest ages, like from 0 to 5 years old, and you took antibiotics when you were a little baby, or you were born with a C-section, or you didn't breastfeed a lot or longer time, or you didn't eat a lot of fibers when you're little, your microbiota is probably not that much robust enough. So the recovery and the resilience from such a cleansing or such an intervention will not be that easy. And this is why you see people, some of them, listen, I actually took an antibiotics and really suffering since then. I couldn't recover. I'm just like sick all the times, although I just took them one time. Go back to history. What happened to your microbiota when you were little? And this is very, very important that I really highly emphasize that it is very crucial that people take care of the baby's microbiota because it's crucial how they will grow up with it. Another example, this person actually took a surgery, abdominal surgery, and you can see that the diversity dropped even lower, like really bad, which also recovered simply because that person probably had a resilient microbiome that allows him to actually recover from that kind of intervention. And this is what I've told you. This is what is very important to go back to that recovery, to that resilience. It is the first 1,000 days, which is one of the most important period of life of an infant from a microbial perspective, where there are a lot of very good things that you can do for the infant and a lot of other bad things that you should not be doing. So including antibiotics, reducing as much as possible. I mean, if a baby have to be born to a C-section, you cannot do it otherwise. Although today, in some hospitals in China and probably some coming actually to Europe, what we're doing today for babies born with a C-section, what we call, we do a vaginal microbiota transplantation to the baby. So basically, before the baby is born, if it's planned C-section, we go to the mother and then we take some vaginal fluids, right? When the baby is born from a C-section, we rub him, like with these vaginal fluids to enable him to acquire those microbes that he would have not encountered this way. So this is, I really hope this kind of practice gets closer to application in other countries beside China and few countries in Europe as well. One of the forms as well of this dysbiosis that is actually the manifestation of many, many different conditions. So there are some factors that can definitely influence the composition of the gut microbiota, which lead to a dysbiosis. And this dysbiosis is actually can be just beyond the gut. So like what happens in the gut don't stay in the gut, it actually goes beyond the gut, right? So we can have the gut skin axis, acne, psoriasis, atopic dermatitis, wrinkling, aging. We got pancreatic axis, got lung axis, got heart axis, got endocrine axis, brain axis, which is one of also the most studied interesting fields of this times. And the other axis that, apologies, I don't see it in the image, the gut eye axis, right? So what happens in the gut also do influence, and there are some reports who actually point to that kind of things. So meaning that don't think that making or improving gut health will only result of improving digestive system. It actually improves just beyond the gut. We call it the beauty from within for the gut skin axis. We call it psychobiotics for the gut brain axis, and so on and so forth. Would we also see from a phenotypic perspective, meaning if there is a dysbiotic gut microbiota, it actually results in somebody of being obese. But that obesity, it's not a result of just the missing microbes or the dysbiotic microbes. It also connects to all the other things that I've just mentioned to you, leading to some complicated conditions to way more complicated conditions. So today we could link cardiovascular disease to obesity to gut dysbiotic gut microbiota. So there is a connection between the events, between what happens in the gut, what can actually lead to way more complicated conditions. So focusing on improving gut health, it definitely is one of the first way to someone should look for when the medication don't work, or maybe as a prevention. Just look at this as a first thing that you probably educate yourself on what is happening in the gut and why it's influencing your life in the other sides as well. I will give you now some example and some case studies. You've probably seen this, although it was a controversial study, really. So this was like a very interesting study where they took twins. The reason why they took twins in this study is because they wanted to get rid of the genetic variabilities. So if they are twins, they have the same genetic background, meaning that, okay, we don't actually care about the genetics in this case, because genetics is also one of the co-founding factors that can influence the variability in the microbiota. And these genetic identical twins, there were 10 men and 34 women. So we want to give them randomly, these people actually, either 21 of them, one twin took a healthy vegan diet, and the other twin was eating a healthy omnivore diet. So a lot of food, some of it was junk as well. And they wanted to see if there was an impact on some parameters. And one of them here was lipoprotein cholesterol, right? And what you could have seen that basically the people who were vegan, they had low cholesterol compared to the people who were omnivores, or low LDL to be precise, right? And in some annex of the study, they showed as well that this vegan eating twin had a higher diversity of the microbiota compared to the people who are actually eating more omnivore food. And this is an interesting study. Why it was controversial? Simply because people, they were actually eating this at home. So they were recording what they were eating. So a better study would have been that these people actually ate at the clinic and all their eating was recorded and probably given by the clinic itself. So it could have definitely been done better. But there is another study who's actually done better. So this study was comparing an Italian cohort with a Dutch cohort, and they were trying to look into the balance between fiber and protein. And they were trying to say, okay, if we have a cohort of people actually eating more protein compared to people eating more fiber, what happens to the gene richness and what happens to the microbiota? What I found very, very interesting that if the protein and fiber ratio, like the Italian males, is high, right? It's like close to 5.1, that they're eating 5 times more protein than they're eating fiber. They had a more proteolytic gut microbiota, which is associated with protein breakdown and animal products. On the other hand, like a Dutch female, which had a lower protein to fiber ratio, they had more fiber degrading gut microbiota. What you need to understand, the microbiota doesn't degrade fiber for fun. They actually degrade it for a simple reason. They degrade it to produce short chain fatty acids, to produce some vitamins, to produce some serotonin, to produce some molecules. They're beneficial and they're very known to be beneficial. Short chain fatty acids like butyrate, acetate, propionate are known to be good for gut barrier function, reducing the leaky gut, good for the brain function, and so on and so forth. So increasing the ratio of protein to fiber will reduce the ability of those fiber degrading microbes to do their job and hence producing those metabolites. So meaning increasing the fiber ratio, right, and giving more fiber to the bacteria could be a very good intervention, right? And this is what you see in the previous study, where the people that were eating more vegan, they had low cholesterol levels, LDL cholesterol levels, right? So it just makes sense that to give more fibers to the bacteria to actually do their job. However, also what I want to mention is that these bacteria, they're also fingerprints, right? So I'm just trying to see the time that I still have like 10 minutes, right? So this bacteria, they're also fingerprints. And this study was appeared like not longer than 2 months ago. And I honestly, when I've read the study, I was just stunned. I actually didn't expect this, honestly, as a scientist. So in this study, they took 86 participants and they took a lot of blood samples where they did proteomics, metabolomics, lipidomics, cytokines, lab tests. They took stool microbiome, skin microbiome, oral microbiome, and nasal microbiome. And they did this over a period of 6 years. They wanted to assess what are the changes that happen to somebody's microbiota, including the different microbiota, and are these changes persistent? Are they shared between the people or not? This is what I found. It's basically after the 6 years, they found that there are what actually influence someone's health is not the microbiota that they share with the other people. Remember I told you earlier, there are some keystone species, some keystone microbes, bacteria that we all share, and there are some that are specific. Someone would think that the ones that will be more responsible of our health are the ones that we do share altogether, because if you got COVID, maybe all the bacteria associated with that will all be influenced. What happens is that the actual bacteria that are more persistent, they're more stable when we get sick, aren't the bacteria that we share with others. They're the bacteria that are more personal to us. It's like a fingerprint. This is what I told you before. Bacteria may be and will be in the future a fingerprint that we could use it to actually tell exactly who you are, right? So we don't actually need your fingerprint anymore. We just need to look into your stool samples or your nasal samples. And this is actually correlates the changes of our microbiota. It also correlates with this image that you've probably seen it, and you will see it in most of the infectious disease reports, is that a lot of infectious diseases correlates with the fact that there's some infectious diseases goes down. It also correlates with immune diseases going up, and which also correlates with the microbiota diversity going down. And this is what I try really to do, is part of my mission. Part of my mission is preserving the human microbiota from extinction. So when you look into populations that humans didn't really make a lot of changes, like the hunter-gatherers, these tribes, they live separate from the civilization, and they don't actually consume a lot of processed food, probably never took any medication for some cases. And you can see here, this is people cohorts from the US. And this is looking into the microbial diversity from this axis. And you can see that people from the tribes, they're way different from people from living from industrial associations. A new study actually also showed that when you take somebody from the hunter-gatherers and you look into their microbiota, and you take somebody from California and you look into their microbiota, you will find that this person from California misses 723 species of bacteria. A new report saying that even 100 years ago, compared to today, we've lost 50% of our microbial diversity, meaning that at some point, our microbiota may not be able to make anything meaningful that will keep us healthy. So it has to be and must be a top priority for you as a human being and for societies to preserve this, whatever is existing, before they go missing. And this is all the diseases that correlates with a dysbiotic microbiota: rheumatoid arthritis, type 1 diabetes, eczema, asthma, IBD, IBS, cardiovascular disease, type 2 diabetes, NFLD, cardiovascular diseases, mental health, autism, and so on and so forth. Either they correlate with the absence of those bacteria that we're missing and missed, or the metabolite they're no longer being able to produce. So fixing those microbes is very important. But this is not only for the gut. Microbiota, as I said, they're way beyond the gut. They're existing in every living thing, including from plants, planets, and us. And one of the other things that are really important to me is that not that much spoken of is the vaginal microbiota. It is why, for a simple reason, I've told you, the infant microbiota is very important, because if you mess up with the infant's microbiota when they're young, from 0 to 5 years old, it might be very difficult to fix it afterwards, really difficult, because you do whatever you do, if the microbes are missing, they're missing, right? Same for the women's and vaginal microbiota. The vaginal microbiota is the first microbiota that the infant gets, not only for the benefits of the infant, but also for the benefit of the woman, because this vaginal microbiota also shifts. And I will tell you in a very, very, how to say, when I speak about it, it actually gives me goosebumps, really, because of the study of its interest. And this is the study I'm telling you about. This was a woman. It was 30 years old. She actually had 5 miscarriages, and she was suffering from bacterial vaginosis. And as you can see in red, this is called Gardnerella. It's a pathogen that is known to cause bacterial vaginosis. And as you can see, each bar here represents the vaginal microbiota composition of this woman. So this is one test, the second test, and so on and so forth. And as you can see, this woman was really suffering from bacterial vaginosis. So what this group did is basically they took a donor where they had a very high amount of Lactobacillus crispatus. It's a very beneficial vaginal microbiota. And they took this vaginal secretion and they transplanted them to this woman. So this woman suffering received a vaginal secretion from another woman, a healthy woman. And as you can see, after the first donation, this woman shifted completely. So shifted from a completely dysbiotic vaginal microbiota, rich with Gardnerella, to a more normal vaginal microbiota. So the vaginal microbiota is actually, it's not like the gut, we're very diverse. The vaginal microbiota is the opposite. It's not diverse at all. It actually mostly dominated with Lactobacillus and Lactobacillus crispatus, Jensenii, and Gasseri. And as you can see, this woman, before the pregnancy, became Lactobacillus dominant. It happened that she had a new episode of Gardnerella, which disappeared because her vaginal microbiota became more resilient. Guess what? This woman became pregnant after 5 months, which is amazing. This was one case study, and there are many other reports that have been conducted, many other clinical trials. And I actually had a chance to interview one of the leading authors of this study, Professor Neda Gioia. So on the podcast, so you feel free to have a listen and get inspired and, how to say, amazed as much as I am with this kind of things. So I hope that you don't stop on the gut microbiome, look beyond, because the microbiota is everywhere. It's also responsible of some very debilitating diseases, including women's health and the vaginal microbiota in this case. But again, you tell me, okay, you told us a lot about the microbiota and what's happening. So, okay, what application do we have today? So the only application that exists today from a therapeutic perspective, I'm not speaking about probiotics, which aren't therapeutic, they're food supplements, so don't mix it up. Therapeutic application, I mean, they're approved by the FDA or the EMA or the Australian authorities. The only one, and it was approved in December 2023, and sorry, December 2022 and January 2023. It's called fecal microbiota transplantation for recurrent C. difficile infection. And there are 3 companies who have managed to do that. One in Europe and the States and in Australia. But someone will tell me, okay, fecal microbiota transplantation isn't that old? Yes, actually it is. So the first fecal successful microbiota transplantation was in 1958, where we've used it to actually cure some patients with pseudomembranous enterocolitis. But it took a lot of time, a lot of investment to get something from concept to actual therapy, and that's how long it should take. And by the way, fecal microbiota transplantation is not 1959. It's actually called the yellow soup from the Chinese medicine. They were using fecal transplants back then to actually cure some conditions like fever and so. But the science had advanced since, and as I told you, sometimes we feel that this is all in new science that we've discovered today, but a lot of it was discovered before. We've actually, in the 72, we knew that microbiota can actually metabolize drug. So today we think that, oh, do they do that? Yes, actually we knew that a long time ago. And we sometimes ignore it. But if you take an anti-inflammatory drug today and you see some people do respond and others don't, it's because maybe their microbiota is responsible of that. So keep in mind that the microbiota is responsible of drug metabolism. Also keep in mind that microbiota do shift in the early ages. We knew it a long time ago. Keep in mind that microbiota, we've managed to transplant bacteria from healthy people and obese people and make the mouse obese, meaning there is a causality in some conditions where we could actually transfer bacteria that makes somebody become obese in that sense, right? We also knew that basically using diet as a medicine, using food as medicine, it also works, and it works through the microbiota to some extent. And this is the lab of Jeffrey Golden that I've mentioned, where they found obese individuals that had reduction in some relative abundance of Bacteroides. And then we could have reversed that obesity using the diet in that sense. So diet is a medicine itself. Sometimes we ignore that antibiotics do harm, but we actually knew a long time ago that antibiotics, imagine if you take an antibiotic like ciprofloxacin, which is not that broad antibiotic, it's more or less small spectrum. It can literally knock off 1/3 of your gut microbiota. 1/3. So I don't know why people do take antibiotics when they just have flu. I mean, don't take antibiotics for flu. Don't prescribe antibiotics unless you're sure it's a bacterial infection. Preserving the microbiota should be a priority while keeping patient safety if they need actually antibiotics treatment. Today, there are a lot of work and advancement in terms of microbiota research, in terms of cancer therapy. Imagine if we know that if you give chemotherapy to somebody, that person may not benefit from it simply because they missing some key microbes. Some studies show that Akkermansia municiphala, which is also used now to reduce obesity, for instance, or increase satiety, it's also responsible for increasing efficacy of chemotherapy. Faecalibacterium prosnitzi also is being investigated into improving the efficacy of checkpoint inhibitors, PD-1, as an example. So microbiota is not just only metabolizing some drugs, but can also improve the efficacy or reduce the efficacy of other drugs. In present, what we have today, we have actually different tools in our plethora. We have some tools that are donor independent. So we could actually isolate some microbes from either food matrices or from healthy donors. We make broad consortium. So we make a little bit of composition of microbes, defined consortium. We can engineer some microbes to actually improve their efficacy. We use probiotics, we use prebiotics, we use postbiotics. So as you know, prebiotics is the food between parentheses of probiotics, and postbiotics as well. A lot of companies, a lot of institutions as well, they're now looking into what are the small molecules that this microbiota is able either to produce or convert from other substances. And then we have some donor-derived. The donor-derived is what I've just told you about, this fecal microbiota transplantation that we're using from donors, or the vaginal microbiota transplantation. There are also some, how to say, case studies where we actually did a skin microbiota transplantation. So one doctor is called, he's from Belgium, he's called Dr. Armpit. He's very famous online. Check him out. He's actually done a skin microbiota transplantation for people who had malodor. So basically took people who had no odor or good odor from the armpit, and then transplanted that microbiota to other people who had malodor armpit. And guess what? It improved their malodor. So interestingly, microbes can do wonders, and if you do them properly. The future is how we can actually transfer these discoveries. All what I've showed you from the 1954, when the first fecal microbiota transplantation was done, and only to 2023 was actually approved, is what are the other things that we can do? Not longer than 3 weeks ago, there was a group from a Dutch university here in Belgium who basically took fecal microbiota transplantation to improve mild to moderate Parkinson's disease in a very interesting study, a randomized clinical trial. And they showed that some people receiving fecal microbiota transplantation did improve from some motor behavior in the Parkinson's disease. In other studies, we showed that autism, it also improved. A lot of companies are actually trying to use this now to cure some specific types of cancer. Fecal microbiota transplantation is one way, but there are other ways of actually using the microbiota to modulate disease. We call them live biotherapeutics. This is where we isolate one microbe, we study that microbe, how it does work, and we try to target specific disease with their specific mechanism of action. This is the future of medicine. If you look into from a business perspective, the microbiota field is one of the fastest growing field, all covering all different fields, including the very famous AI field. So meaning that there is a future to this. However, we have to be careful. What are the things that we can use today to, how to say, to transform and change people's life to the better? What are the things that we should add more science, add more data to be accurate, to be precise, and don't give false hope? But I think there is a very good hope in terms of using microbiome and microbiome therapies to actually improve people's life worldwide. And yeah, I think that was my last slide. If you're interested to know more, feel free to follow me on LinkedIn and or reach out via email or definitely through the Ocular Wellness and Nutrition Society. Pleased to be here with you, and I hope I've managed to get you something.
I am so the wealth of information, and I know a lot of our audience out there is going to be really interested to knowing more about what you talked about today. It's such a widely necessary subject matter that we lack, as you know, the foundational education. And I am so thrilled that people like yourself are doing this for us. So I would like to go over some Q&A because there's a lot of questions, as we know. It's just the problem is, you know, in a lecture for 1 hour, there's only so much you can squeeze. And I already predicted some questions, and we spoke about this earlier.
Nice.
So I want to get the one that is obviously out of the way. The probiotic question. What's the best probiotic? And I know we can't talk about name brands, we discussed, but you did give some great indications of what to look for. So let's talk about that, because that's a very highly used question here.
Absolutely. I think, again, so it is very important to not give science to people, just tell them, okay, the microbiome is great, the microbiome is doing this, the microbiome is doing that. But it's very important to also give them solutions to fix it. I mean, if they have a problem. And one of the solutions, obviously, that we're using today, and that is, how to say, very publicized, that probiotics aren't really like existing. I mean, they exist for a long time, right? But there are different forms of it. I mean, if you look into buy food supplement as a probiotics, you have to be very careful what probiotic you should be choosing. And number one, I always recommend people on when they should be taking probiotics. So some people actually take them at the evening or at lunchtime. Although we know that the stomach acidity is high or is low, right? So if you take probiotic, remember these are live microbes. Some of them, they don't actually resist acidity. Some of them are put in capsules. They don't actually resist that acidity at all. So number one, I recommend people to take probiotics early in the morning before they eat anything, where the stomach is empty. So the probiotics can transit quietly to the intestine. And they don't have that much of acidity, because the acidity of the stomach increases a little bit to get around like 5.86 before the breakfast. So number one, take them early. Number two, don't take any probiotics that probably you don't look for what clinical trials have been done on it. So number one, just look into the probiotic, look into the strains. So not all bacterial strains are the same. I would highly recommend people taking probiotics with different strains and to rotate the strains. Meaning, if you took a brand today that had Bifidobacterium, Bifidum, and Lactobacillus, whatever name on it, try to change the brand or the same brand, but different strains. Rotating gives better ability to this bacteria to engraft, like to stick a little bit more in your intestine. Just rotate those bacteria. Number three, try to get the names of the bacteria and just Google them up. Did they do any clinical trials? I highly recommend people not to take probiotics. We just list the name of the bacteria, just saying Bifidobacterium bifidum, full stop. No, this is okay. It's like I'm saying Dr. Amine, and stop. Okay, what's his last name, right? So Bifidobacterium bifidum NC31. That's a strain name that you can Google and find, right? Bifidobacterium bifidum is like I'm telling you, listen, there is a country. Okay, what country, right? So always look for strain names that are complete, not short, which will enable you to actually research those probiotic names and make sure that there are at least some clinical studies on it. So Google it, you'll find it. And I hope in the future that I know there's some people working in a form of, how to say, scientifically proven databases. There are some databases existing out there which enable you to look, okay, what probiotics are good for what, right? But I hope there will be others where literally you just put your name, put your conditions or your metrics and metadata, which enable you to get whatever you should be taking afterwards.
Wonderful. And that is strain specificity is very, very key.
Very important.
For purchasing probiotics. And I always say probiotics are very different than supplement purchases as a whole. It actually has its own kind of like checkpoints, right?
Absolutely.
So that brings us to another actually good question. And I kind of have my own mini answer, but I'd like to see your opinion. When we do order probiotics, how do they survive extreme temperatures like transportation, et cetera? So when you get point A to point B, because these are live things, right? They're alive technically.
Yeah. So theoretically, probiotics, they should, I mean, at least how it should be done. The manufacturer should do a stability test, meaning that they actually put them at 4 different temperatures. They put them at 4 degrees. They put them at 24 degrees, at 25 room temperature, and they put them at -20, and they put them at 40 degrees. And this is what we call them a stability testing, to assess their stability over time. Theoretically, they should lose at least 2 logs at 40 degrees up to 6 months, meaning after 6 months, they will be losing 2 logs, meaning if they were like 1 billion, they become like 1 million, right? So that's 3 log actually, or 10 million. So if the manufacturer did not do a stability test and they did not pack them properly, very likely they'll die. But for some cases, if the probiotics are heat resistant, like spore forming, you can ship them wherever you want, they will not die, right? But the packaging should play a role. I highly recommend people as well to check whether the manufacturer actually did any stability testing on them, and they should mention on their packaging they're heat stable. If they didn't, obviously they're probably dead when they get to your place.
Yeah. No, it's again, all these label reading is a science. One other concept that we discussed that I said there's definitely going to be questions about is the ocular relationship of twofold. One is general microbiota dysbiosis, et cetera, with ocular inflammation, which I personally believe that there is a sound connection. And then number 2, the ocular microbiome, which we discussed, we might have a part 2 to this because of how unique that conversation and research is. So if you want to just kind of touch on that for the audience as well.
Absolutely. So one of the pictures I mentioned there is we'll call it the systemic microbiome. It doesn't mean that bacteria are swimming in the blood, but it means that their molecules they're producing, they swim in the blood. Just to give you an example, communication between the very obvious one, the easiest one, is the gut-brain axis, where the communication happens with the vagus nerve. So metabolites they've been producing in the gut, they crosses the gut barrier, they go to the blood, and they transit to the brain through the vagus nerve. And this is what actually increases, or reduces some brain activities. We don't actually know how these molecules reaches the eye, although there are evidences that inflammation in the eye depend to some extent to the inflammation in the gut. So if you have a dysbiotic gut microbiota, there are very few reports that showing that dysbiotic microbiota can cause some eye issues, right? The other side of the curve, if I may say, is this eye microbiota. The difficulty today in the eye microbiota is its study. So how do we study the eye microbiota itself? Meaning, how can we isolate the eye microbiota? In the case of the gut, is easier because you just get stool samples. The skin, you just get the swab. The vaginal, similar. With the scalp, is similar. You get a swab. With the eye microbiota, it's a little bit difficult. So the field is growing. And I hope that maybe in the future we'll be discussing more about it. Like, how can we study the eye microbiota without contaminating the eye itself, right? Because the contamination is the issue here. Are these bacteria in the eye, are they really in the eye, or are they coming from somewhere else? And this is still a debate there, right? However, we do know that at least from a gut-eye axis, what the gut is communicating to the gut, is there are evidence that actually happens, and it passes through different routes, including systemic route, which is through the blood and modulating that inflammation.
Yeah, and that makes sense. You know, the research is still catching up. And we can kind of
Absolutely.
look at it more as a bird's eye view. And when we look at inflammation as a whole in the body, and if it is affecting the ocular system, then there's obvious reasoning to maybe reduce the inflammation as a whole body system versus just think of the eye as the cause, you know, as the end point. So it makes a lot of sense. Along the lines of probiotics, kids versus seniors, in terms of what type of probiotics you would look out for. You know, do we add bifido when we're older? What would you say?
Yeah, that's a very, very important question. Obviously, as I told you, the gut microbiota for infants is very, how to say, weakly diverse. Like, it doesn't have that much diversity. And there is a very recent report who showed the exact strain names that actually someone needs to have in an infant, and someone needs to have as an adult, and someone to have as a senior. So it's very important to pick the probiotics based on the age, because it's very dependent. So for an infant, it's very highly recommended to give an infant Bifidobacterium infantis, because it's one of the bacteria that they would find as an infant. Bifidobacterium longum is not that much prevalent in infant microbiota, is more prevalent in adults. So adapting the probiotics to the age is very crucial. That's why when you pick the probiotic, you must look whether that bacteria was actually, or probiotic, previously used in infant or discovered in infant microbiota, or it was just an adaptation from an adult microbiota.
And, you know, more of, I'm going to kind of blend a question, and this is kind of a personal concept that I think, do you believe in stool profiling and microbiome testing right now as a, you know, as a general standard to perhaps be able to navigate that precision that you're talking about? Is that are we there yet?
It's another debate, right? So I'm not, so there have been a lot of, how to say, a lot of debate on whether this microbiome testing is something that people should be doing. Is it just like money grabbing and marketing, or is it just really something that can actually do something? It depends on what technology they're using and how much they want to share, right? So if someone is using 16S, which is just sequencing the 16S gene, and then telling you, listen, we actually know exactly what's happening in your gut, and we're going to give you nutritional advice and buy our supplements, don't do that. Because it's very likely I'm just grabbing you to buy the supplement. The 16S microbiome testing is probably providing nothing, just high level. It's like I'm flying with a helicopter on a city and just listen, there are people living here. Okay, but who are they? What they're wearing? Their age and what's so. When we get into metagenomic testing, literally knowing the species, what are they, what they're doing, probably even metatranscriptomics, what they are doing. This has become more interesting when they actually can put some actionable information that people can actually use for recommendations, either probiotics or nutritional supplements and what's so. So what I would advise people usually to find companies that don't just do microbiome testing, because a microbiome as a standalone is not an isolated organ. The microbiome lives in another organ, which is the human body, which is connected with the systemic. I highly recommend people to do microbiome testing along with a blood testing, because this will enable you to assess what is there, what they're doing. And then you could make recommendations based on that. So if you buy your microbiome test separately because you couldn't find one doing it both, try to get a blood test in the same time and ask them to do some predictions on what you should be doing based on that information. This is my advice, at least, that a microbiome test today, with the information we have, probably we still need more to actually make actionable advice, although it's still useful to some cases. If you have a healthy microbiota, maybe it's not that much important. If you have a very diseased microbiota like dysbiotic, it can be very, very obvious. Like I had a patient who basically sent me 2 microbiome tests done in 2 different companies. Both the microbiome tests show that this person had 5 keystone species that are gone. Didn't have any bifidobacterium, didn't have any lactobacillus, didn't have any Ehalibacterium and whatsoever. And she was diabetic, pre-diabetic, suffering from depression and obese. It was like very obvious that this lady needed different type of probiotics and different type of prebiotics, and it was fixed afterwards, like 5 months later, right? It's not like a quick fix and you press the button and it's gone. But this is very isolated cases that need really thorough understanding of the microbiota and its diversity. And a microbiome test will never give you that. They just give you a result and they tell you eat that or don't eat that. And this is not what you should be doing. You should be at least have actionable advice.
That makes a lot of sense. Use it as a complement to the whole story.
Absolutely.
Last question, I'm gonna kind of blend 2 questions. Foods that help your microbiome, as we know, there are cultured foods that are very supportive of microbiome health, et cetera. And is that a way, how do we build that when somebody has like a colonoscopy or colonic cleanse? Do those things help?
So what I will always say to people, number one, this is not a medical advice. Always ask your doctor. I'm a doctor, but in biology, microbiology. So I'm not going to give you medical advice. Don't push me to do that. I will never do that. However, your doctor should be able to advise you. There are some foods that depend on your condition that will definitely increase your microbial diversity, and they aren't cheap. They aren't expensive, sorry. So one routine that people should be taking, if they can tolerate that, kefir or fermented foods, right? They're cheap and they can definitely increase your microbial diversity. Prebiotics or prebiotic food like resistant starch, for instance, is a mean to increase your microbial diversity. So, and very importantly, I think people focus a lot about what they should be taking instead of what they should not be taking. So eventually, if you stop taking a lot of what you should not be taking, like a lot of processed food, a lot of food that contains preservative and junk food and what so, and eating sugars, not sleeping very well. Sleep can influence the diversity of microbiota. Not doing sport, maybe you should start in doing sport, right? Like it's a compounding factor. It doesn't like just come like this. But I would say, number one, make it easy to yourself. Stop doing things, the easy thing. So number one, you just like keep the money in your pocket because you're not spending it on something that you should not be eating in the first place. And after that, in the meantime, add some fermented foods that you can tolerate, right? Some fibers increase slowly. If you have IBD or RBS, maybe consult with your doctor. But there are some fibers that IBD can tolerate. So just do it slowly, build on it, and then you find your balance. It's all about the balance. I'm not telling you stop eating meat. No, keep eating meat if you like meat, but don't make meat your main course and your dinner and lunch and breakfast, right? Just keep it in balance. As we said, protein, fiber ratio does matter. So keep a balance. The best thing that you should be doing if you want to get into this, teach yourself about Mediterranean diet, like tons of millions of studies showing that Mediterranean diet is a very good diet to increase your microbial diversity. Educate yourself on that. Trust me, you will probably need nothing else except just sticking to your Mediterranean diet.
Yeah, it sounds easy, but it's
not that easy.
yeah, and that's why we have all these issues, right? So
yeah.
So I'm not going to take up more of your precious time, but I do want to thank you personally to Dr. Zorgani and his awesome initiative with the Microbiome Mavericks. You can find him at the website microbiomeMavericks.com. And I thank you for all the attendees today and the future attendees with our recording, because I know this is a tough time of day, especially if you're working. And don't forget to visit the Ocular Wellness and Nutrition Society website for how you can be a part of our mission as to bring this education to our professionals. So thank you. A happy Thursday. And yeah, I hope we have more conversations in the near future, Dr. Zorgani, about this really, really important topic.
My pleasure. Really my pleasure. Thank you so much for having me. And yeah, thanks for the listening.
All right. Thank you, everyone. Have a good
Take
day.
care.
All right.
Cheers. Bye.